Thank you, sir. I wish everyone who worked with me was as easy to get along with as Professor Durson. Thanks very much to Masoud and the organizing committee for inviting me to this beautiful venue for a conference. It's been great scientifically and socially. Thank you. Today I'm just going to give you a brief overview of neuroactive steroids. There's been a lot of work done on them in recent years, but I'm going to focus today on schizophrenia and depression and mention a little bit about anxiety disorders. So objectives here, what's meant by the term neuroactive steroids. I'm also going to show how neuroactive steroids may be involved in the etiology and pharmacotherapy of schizophrenia and depression. So, neuroactive steroids are steroids that exert they can either originate as circulating steroid hormones, or they can be synthesized in the brain itself from cholesterol, in which case we call them neurosteroids. So the two terms are used interchangeably, but there is a slight difference. These neuroactive steroids may act at receptors for several neurotransmitters. As looked at their allosteric modulation of the GABA-A receptor and the NMDA glutamate receptor. So these two very important neurotransmitters, an inhibitory neurotransmitter, excitatory neurotransmitter. As we all know, the balance between the two in the brain is very important. So some of the neuroactive steroids act as positive and some as negative allosteric modulators at GABA-A or NMDA glutamate receptors. Some of the neuroactive steroids have been reported to have anxiolytic, antidepressant, and or neuroprotective effects. Some of them have all three of those. They appear play a role in neurodevelopment and in physiological processes, including sleep and cognitive functions. Here's the typical structure of some of the neuroactive steroids. They have that typical four-ring conjugated structure. These are the main ones that we deal with. There are a large number of them, but allopregnanolone, TDOC, DHEA, the sulfated form of DHEA or DHEAS, pregnenolone, and pregnenolone sulfate. And those are the ones that I'm primarily going to talk about. There's a P450 side chain cleavage enzyme to form pregnenolone. And you can see the center role here that pregnenolone plays subsequent formation of a number of very important neuroactive steroids. So the pregnenolone can be sulfated, it can form here to get to these two very important neuroactive steroids, allopregnanolone and TDOC. So as I mentioned, neurosteroids are synthesized beginning with cholesterol. Formation of pregnenolone from cholesterol is regulated by the translocation protein TSPO. This was formerly called the peripheral or mitochondrial benzodiazepine receptor. This favors the transport of cholesterol from the outer to the inner mitochondrial membrane, initiating synthesis of neurosteroids. Pregnenolone is then converted to a variety of neurosteroids, as I've mentioned in the previous slide. Just to go over some of the individual ones and how they differ in their pharmacological actions. Allopregnanolone, which has probably been the most frequently studied of the neuroactive steroids, acts as a positive modulator at the GABA-A receptor, and it's a very potent. This is a very potent effect, providing anxiolytic action. So we're talking about a compound that's really as strong as the benzodiazepines. Progesterone is also a positive modulator here. DHEA is a positive modulator at the NMDA glutamate receptor. The sulfated forms of DHEA and pregnenolone are negative modulators at the GABA-A receptor and positive modulators at the NMDA receptor. And pregnenolone itself, which remember plays a very central role in synthesis, is a positive modulator at the NMDA receptor. And I'll talk a little bit more as we go along about how those fit into their actions. The HPA
HPA ekseni bildiğiniz gibi
particularly in mood disorders and stress-induced release of neuroactive steroids, again, particularly allopregnanolone and TDOC, the ones that are very strong modulators at the GABA-A receptor, may represent an endogenous homeostatic mechanism for restoring the GABAergic system and subsequently restoring normal activity of the HPA axis. We know that GABA-A receptor activity can inhibit activity of the HPA axis, which is overactive in some depressed patients. The other thing that's important here is the interaction with brain-derived neurotrophic factor, or BDNF. It may also be involved in interactions of neuroactive steroids with the HPA axis. BDNF, as we know, is involved in regulation of the HPA axis, and the effects of several neuroactive steroids on the activity of that axis appear to be related to their effects on BDF content of the hippocampus and the hypothalamus, suggesting that BDF is involved in neuroactive steroid regulation of that axis. Okay, I'll deal now with schizophrenia first, and give you some examples of how the neuroactive steroids may be important. Allopregnanolone has been reported to have antipsychotic-like and anxiolytic effects in several animal models. The antipsychotics olanzapine and clozapine increase allopregnanolone levels in rodent brain, and this may counteract the reported decreased GABAergic activity that occurs in schizophrenia. Pregnenolone, that's a very central neuroactive steroid in the synthesis, and pregnenolone sulfate enhance learning and memory in rodents at concentrations known to be present in human brain. The atypical antipsychotic clozapine increases hippocampal levels of pregnenolone, possibly by enhancing binding of that translocator protein involved in the formation of pregnenolone from cholesterol. Most of the work here looking at levels of pregnenolone with the atypicals has been done with clozapine. But in fact, I think other atypicals probably have a similar effect. We've done some work with ziprasidone and found that it also increases pregnenolone levels in rat brain. Christine Marks in the U.S. has done a lot of work on clinical trials with neuroactive steroids, and one that she did with pregnenolone indicated that it may be useful for treating negative symptoms of schizophrenia when added to atypical antipsychotics. And she felt from the preliminary results that they had, it appears to be worthy of further investigation for treatment of the cognitive symptoms as well. Pregnenolone is metabolized to pregnenolone sulfate, and this is, as I mentioned before, a positive modulator at the NMDA glutamate receptor, and it's also metabolized to allopregnanolone, which is a positive modulator at the GABA-A receptor. So what might be happening in these clinical studies when you administer pregnenolone is that you're counteracting the functional deficiencies of both glutamate by increasing pregnenolone sulfate levels and counteracting the proposed GABAergic hypoactivity that's been reported in schizophrenia by forming allopregnanolone. So by giving pregnenolone, you formed two other steroids, one of which is a strong positive modulator at the GABA-A receptor, and one that's can counteract the glutamate deficiency or the NMDA deficiency. Neuroactive steroids in schizophrenia, as far as animal models, in this conference so far, we haven't really talked very much about dopamine, which is unusual for a schizophrenia conference, but we shouldn't forget that dopamine probably has an important role as well. And in a mouse model of schizophrenia, the dopamine transporter knockout mouse, pregnenolone administration reduces hyperlocomotion, the stereotypic bouts, and pre-pulse inhibition. So in a mouse model, it does appear to help with the dopaminergic hyperactivity as well. So we shouldn't forget that the neuroactive steroids could be affecting dopamine as well as GABA and glutamate. DHEA itself has anxiolytic, antidepressant, and anti-glucocorticoid properties, and when administered exogenously may reduce negative affect. When it's administered to schizophrenic patients, it reduces activity in the amygdala and hippocampus, but also, and Christine Marks, her group has shown this, it also enhances connectivity between these two brain areas. And it also increases activity in the anterior cingulate cortex, which may affect cognition. There's also considerable evidence that schizophrenics have lower plasma levels of pregnenolone and DHEA sulfate, that should be a capital S, and elevated cortisol to DHES molar ratios compared to normal controls, and this is work that Dr. Ritzner has done in Israel. If we go on to mood disorders, focusing on depression, CSF levels of allopregnanolone have been reported to be decreased in patients with major depressive disorder, and you can correct this by giving a number of antidepressants, including fluoxetine, mirtazapine, etc. And actually, with the SSRIs, this effect on elevating allopregnanolone is stronger than their effect on inhibiting serotonin reuptake. This is work that was done in Mimo Costa's laboratory. Socially isolated mice show increased aggression, fear, and anxiety, and these are associated with decreased levels of allopregnanolone in corticolimbic areas of the brain. And fluoxetine and its active metabolite norfluoxetine can reverse that allopregnanolone decrease and the behavioral dysfunction that occurs, again, as I said, at lower doses that are even than those required to inhibit serotonin reuptake. Fluoxetine and olanzapine, which is, as you know, is an atypical antipsychotic that also has been reported to have antidepressant properties, increased levels of pregnenolone and its metabolites, again including allopregnanolone, in rat hippocampus. The SSRI antidepressants and mirtazapine have been reported to increase levels of allopregnanolone quite markedly in rat brain, although there probably should be more comprehensive acute and chronic studies done here for comparisons. Carrying on with depression, it's been reported that DHEA has antidepressant-like effects in humans and animal models. However, if we look at serum levels of DHEA or sulfated metabolite, they've been reported to be reduced in humans after chronic treatment with a number of antidepressants. So there's a bit of a disconnect there that doesn't quite seem to fit. Anxiety, not as much work's been done with the neuroactive steroids and anxiety disorders, but because of the close connection between anxiety and depression, I'm just going to mention this briefly. The involvement of neuroactive steroids in anxiety disorders, again, mainly involves allopregnanolone and TDOC, these very strong GABA-A receptor modulators. They have affinities at the GABA-A receptor, as I mentioned before, equivalent to or even more potent than the benzodiazepines. So positive and negative modulation at the GABA-A receptor is strongly associated with anxiolytic and anxiogenic activity, respectively. We've looked at, or my colleague Jean-Michel Lamelodo in Edmonton has looked at a number of anxiety disorders, and in fact, in all of them, we did see abnormal levels of neuroactive steroids, but each of these disorders had a different pattern. So PDSD showed increased plasma DHEA and its sulfated metabolite and decreased CSF allopregnanolone. Panic disorder increased plasma progesterone, DHEA, pregnenolone, and TDOC. And when we challenged these patients with cholecystokinin or analogs, they, unlike normal controls, you didn't get an increase in allopregnanolone. So they don't seem to have that ability to increase allopregnanolone levels to counteract the panic attack. PMDD, there was decreased DHEA and pregnenolone, and generalized anxiety disorder and social anxiety disorder, both of those had decreased plasma pregnenolone sulfate. But we were quite surprised to see that there were quite different patterns in all of the panic disorders. So, future directions for neuroactive steroid research. We need to do more studies on glial cells, and that applies to almost every area of biological psychiatry and neurochemistry. We know that glial cells far outnumber neurons, and they're not just there to support neurons, they have other functions as well. Glial cells, particularly astrocytes, possess the ability to form neuroactive steroids, and they may be important in the glia neuron and glia-glia crosstalk. Just a diagram here of glial cells. Carrying on future directions, we need more studies on the effects of the currently available antipsychotics and antidepressants on levels of neuroactive steroids and the enzymes that are involved in their synthesis and catabolism in specific brain areas. We need more studies on the effects of these TSPO ligands, which affect translocation of cholesterol, and also more synthetic analogs of neuroactive steroids. Some of them have very short half-lives, and we're now looking at preparing some analogs. The other thing is when you're working with neuroactive steroids, the more specific and sensitive equipment you have to measure them with, the better, because it's if you can measure several of them at once, it's going to give you much more information than just focusing on one or two at any one time. Of course, plasma levels of neuroactive steroids may differ markedly between the follicular and luteal phases of the menstrual cycle, and researchers should be taking this into account when they do design their studies. So, in summary, levels of neuroactive steroids in the brain and body fluids are altered markedly in a number of psychiatric and neurological disorders, and by drugs that are used to treat these disorders. Today I've focused on schizophrenia and depression, but we know there are abnormal levels or metabolism of these compounds in MS, which has usually a number of psychiatric disorders associated with it. And we've shown this in animal models and in human studies, that you do get a decrease in the activity of a number of the enzymes involved in synthesis of the neuroactive steroids. The same is true in anxiety disorders, which I mentioned, Alzheimer's disease, and hepatic encephalopathy, which we've also done a lot of work with. So the neuroactive steroids may be important markers in a variety of disorders, but also may represent future drug targets and even drugs themselves if we can make the appropriate analogs. Acknowledgements, just the funding from the various Canadian sources, and my colleague Jean-Michel Lamelado, Chris Power, Samir Abuka, Nick Mitchell, Gail Rao, Serdar Dursun sitting over there, Catherine Todd, and Aaron McKenzie. Thank you.